Thanks everyone for coming, and I'm honored to be the first one to give an inaugural lecture in this brand new auditorium. It looks really nice. So, as they kindly introduced, and I'm interested in species-specific biological time. I'm interested in why my time is different from other animals' time. And this project or this question started almost 20 years ago when I was a high school student back in Japan. So more than 20 years ago, I read this book titled An Elephant Time and Mouse Time, written by Tatsuo Motokawa. So this very nice book taught me that different animal species live at completely different time scales. Or for example, as you know, the gestation period, our pregnancy lasts for about 9 months, but it's only 20 days for mice. And it takes almost 2 years for elephants. So it's really hard to be an elephant mother. Pregnancy lasts for 2 years, no? 9 months is, I think, enough. Anyway, and the lifespan, we larger species tend to live longer than smaller species. And the cardiac cycle, our heart beats about 60 times per minute, but the mouse heart can beat more than 500 times per minute. And then the elephant heart beats only 20 times per minute. So I was really fascinated by these facts. My time is different from their time, and I wanted to know the mechanism. But first, let me focus on mouse and human developmental time. And as you see, and I will talk about other species later, but I want to focus on mouse and human first. So actually, mouse and human embryonic development are very similar. For instance, this is day 10 mouse embryo, which is almost the same as day 20, 30 human embryo. So the morphologies are similar, the sizes are similar at this stage, and the developmental mechanisms are, in general, very well conserved between mice and human early development. However, as you may notice, the progression speed is different. So generally speaking, human early development is maybe 2 to 3 times slower than that of mice. So that's the question I was really interested in. Why is human development slower than mouse development, even though our developmental mechanisms are very well conserved? However, the entire early development is a bit too complicated for us to study. No? There are too many factors involved, and also it takes too long. I do not want to wait for 60 days for one experiment. That will be a nightmare to my lab members. They wouldn't like it. So I needed a faster, much simpler model system to study developmental tempo. And that's why we focused on this particular segmentation clock as a model system. And as you may know, the segmentation clock is basically an oscillation during development, this rhythm of gene expression. So this very nice movie was taken by Ryo Kageyama's lab, and we are looking at the tail tip of E10 mouse embryo. So during this stage, around E10, embryonic day 10, several genes are known to show oscillatory gene expressions, and this particular gene, HES7, is just one marker gene of the segmentation clock. And as you see, the yellow signal, the expression level of this gene, HES7, is going up and down and producing a rhythm. And also, there's a wave, traveling wave from the tail to the head part of an embryo. So this wave and oscillation are called the segmentation clock. And this rhythm of the oscillation is very important to regulate the timing of our body segment formation. So come to think of it, our body segments, like vertebrae and then our ribs are periodic. They are repetitive structures, right? The same unit repeats several times. They are repetitive because those ribs and vertebrae are derived from these body segments called somites. The somites are those tissue blocks, bilateral tissue blocks. And these somites are repetitive because they are formed one by one repetitively according to the oscillation of the segmentation clock. So this oscillation, the segmentation clock, is the basis of our periodic body structures like ribs and vertebrae. It's really important, but for now, just remember that the segmentation clock is an oscillation during development. And it's a really nice, convenient model to study developmental tempo. But unfortunately, the problem is we cannot do this nice live imaging with human embryos. That's ethically challenging, no? So that's why we needed to recapitulate the human segmentation clock by using human stem cells. Mitsuhiro Matsuda, a postdoc in my group, collaborated with Jantash Aleph, Junior Toguchida, Makoto Ikea at Kyoto University, and they treated human iPS cells, human stem cells, with a cocktail of growth factors and inhibitors to induce human segmentation clock stage cells. So in this movie, I think we are looking at about 10,000 cells, and we are looking at the expression level of this HES7 gene reporter. And we can see nice oscillations of the expression. The signal goes it's pulse, 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 so it's oscillating. It's also a traveling wave from right to left in this movie, just like the traveling waves from bottom to the top part of an embryo. So this way, we can recapitulate the human segmentation clock by using human stem cells. And actually, so this is like a 2D cell culture, but we can make even more realistic 3D segmentation clock by using human stem cells. So in this case, we actually made an organoid, human organoid, by making an aggregate of human stem cells, and then treated the aggregate with a cocktail of growth factors and inhibitors. Then this aggregate of stem cells started elongating, and then these somite-like structures, pairs of somites were formed one by one periodically in a spontaneous manner. So those are the somite-like structures. I mean, I always think this movie looks like a weird organism, but it's actually a man-made object, and it's not a living organism. But it's amazing that these stem cells can self-organize into these spontaneously forming structures. But another good thing about these 3D organoids is that we can observe nice human segmentation clock, oscillations of the segmentation clock as well. Here, the green signal is going up and down, so we can see the rhythm of the segmentation clock. And we can also see the traveling waves from bottom to top. And according to this rhythm of the segmentation clock, those somite-like structures, body segments, are formed one by one. So in this organoid, the somite structure formation is coupled with the rhythm of the segmentation clock. And I guess this model is closer to the actual segmentation clock in actual embryos. So this is mouse embryo, and this is human organoids, and they look relatively similar, and it's very pretty. And I really like this, but for the purpose of measurements and manipulation, actually 3D organoids are relatively difficult. And for the rest of the talk, I will focus on our 2D models of segmentation clock, because the measurements, those 2D models are much more compatible with the accurate measurements. So yeah, this is our typical 2D in vitro segmentation clock model. And on the left is the segmentation clock recapitulated from mouse stem cells. And then on the right is human segmentation clock. And as you may notice, the tempo is different. So in the mouse segmentation clock, the reporter signal goes up, down, up, down, up, down, up, down, relatively quickly. But in the human sample, the signal goes up, down, more slowly. So actually, in the mouse segmentation clock, we found that the oscillation period is typically 2 to 3 hours. This peak-to-peak distance is 2 to 3 hours in the mouse segmentation clock. But if we use human segmentation clock, this period, this oscillation period was 5 to 6 hours. And these values, 2 to 3 hours and 5 to 6 hours, are consistent with in vivo mouse and human segmentation clocks. So this is not an in vitro artifact. So these are physiological periods. And then this is the question I really want to address. Why is human segmentation clock 2 to 3 times slower than that of mice? So I guess this question is much more approachable as compared to the entire lifespan or the entire early embryonic development. And it's much quicker, 2 to 3 hours and 5 to 6 hours. So this is a question I want to address. But before addressing that, let me just briefly explain the mechanism of this oscillation of the segmentation clock. And it's very simple. So you need to remember only one gene name, HES7. This is a core gene of the segmentation clock. So it's a transcription replacer. And this HES7 protein is produced through transcription, translation, and intron splicing. And then HES7 protein is degraded, just like any other genes. But however, this HES7 gene is a transcription replacer which directly inhibits its own promoter. So basically, this is a negative feedback loop of HES7, one-factor negative feedback loop of HES7. And such a negative feedback loop can give rise to oscillatory gene expressions when the parameters are right. So this is a mechanism of the oscillation of the segmentation clock, and the mechanism is common between mouse and human. But then what we found by comparing the mouse and human segmentation clocks is that these reactions are slower in human cells as compared to mouse cells. So this was a collaboration with Jordi Garcia Ojalvo and in Barcelona. So we modeled this one-factor negative feedback loop with very simple delay differential equations. So this part, and the first one represents HES7 messenger RNA, and then the second one represents HES7 protein amount. And if we use these delay differential equations, we can say that there are two important parameters that determine the oscillation periods: degradation rates and delays. So degradation rates means the degradation rates of HES7 messenger RNA and then HES7 protein. And delays means the time delay due to HES7 transcription or intron splicing or translation. So because these two parameters, degradation rates and delays, determine the oscillation period, we measured these parameters both in human cells and mouse cells. And then we found that all the reactions are slower in human cells as compared to mouse cells. So for instance, we measured HES7 protein degradation rate, messenger RNA degradation rate, and they were slower in human cells. And similarly, we measured the delay due to HES7 transcription translation that was longer in human, and then the delay due to HES7 intron splicing that was also longer in human cells. So overall, all of those four parameters were 2 to 3 times slower in human cells as compared to mouse cells. Then by using these measured parameters, we simulated, maybe I will explain and I will show one example of our measurement. because I like showing the actual data. So this is a measurement of protein degradation rate, this parameter. And here we use same exogenous reporter to measure Hes7 protein degradation rate in human cells and mouse cells. But even though we used the same reporter gene, this Hes7 reporter was degraded much faster in mouse cells as compared to human cells. So this is actually not the sequence not due to the sequence difference between mouse and human Hes7 gene. And we actually once swapped Hes7 sequences between mice and human genomes. And this swapping didn't change the oscillation period. This means that the sequences, Hes7 sequences, are interchangeable between mice and humans, but it's the cellular environment that differs between mouse and human. So for some unknown reasons, in human cells, Hes7 protein degradation becomes 2 times slower. So similarly, we measured the other 3 parameters, and all the reactions were 2 to 3 times slower in human cells. And by using those measured parameters, and we simulated the actual oscillation period of the segmentation clock. And if we use human parameters measured in human cells, those values, the simulated oscillation period was 5 to 6 hours. But if we use mouse parameters measured in mouse cells, then the simulated oscillation period was 2 to 3 hours. And luckily, these 5 to 6 hours and 2 to 3 hours were consistent with the actual human and mouse segmentation clock periods. So therefore, we concluded that the human segmentation clock is 2 to 3 times slower than that of mice, because all these biochemical reactions are 2 to 3 times slower in human cells as compared to mouse cells. So the protein degradation, the transcription, translation, and intersplicing of Hes7 gene are 2 to 3 times slower in human cells, and that leads to the 2 to 3 times slower human segmentation clock as compared to mouse clock. So that was our first conclusion. So kinetics, the reactions are slower in human cells than mouse cells. So that's our conclusion. Then we wondered, what about other species than humans and mice? And what about other genes than Hes7? And then what's the cause in the end of the slower kinetics in human cells? So I'd like to address these questions one by one. First, what about other species than humans and mice? And this is a fun part of stem cell work. So this is Jorge Lazaro, one of the PhD students who took the lead in this project. He and I wanted to compare the segmentation clocks of humans and mice and rabbits and marmoset monkeys, cattle, and even rhinoceroses. But it's not practical to keep these animals in the lab, right? But since the rector is here, for the record, I'm saying that I would be happy to have rhinoceroses in the TUD campus. I wouldn't prevent you from making a rhinoceros facility, but yeah, so let me know if any space is available. But even if we can keep rhinoceros colonies in the TUD campus, rhinoceroses are critically endangered, so it's criminal to use their embryos. And therefore, instead of using actual embryos, we obtained stem cells of those species to set up the stem cell zoo in the lab. So this is human iPS cells, human stem cells. And these are mouse stem cells, mouse ES cells, and rabbit stem cells, marmoset cells, and then even rhinoceros cells, and then this is bovine stem cells. So we obtained these animal stem cells from several wonderful collaborators. I probably cannot tell which cells belong to which species, but my lab members can easily tell, wow, this is human or this is rhinoceros. To them, it's obvious, but I struggle to distinguish them. Maybe, yeah, the easiest to me is bovine. It's always scattered, so it's easy to tell. But anyway, this is our current stem cell zoo in the lab, and we are still expanding. I think we now have about 10 species in the lab. And they are the zookeepers. So Jorge Lazaro and Maria Costanzo joined PhD students with Vicas Trividi's group in Barcelona, and they cultured rhinoceros stem cells, and then, for instance, and then induced the rhinoceros segmentation clock cells, and then measured the rhinoceros segmentation clock period. And they did the same for 5 other species, and then they managed to compare the segmentation clock periods across 6 species. And then these animals showed really species-specific oscillation periods, as you see. So the mouse and rabbit cells showed 2 to 3 hours oscillation periods. And then the rhino and then cow showed 4 hours period. And we humans have 5 to 6 hours period. And then the marmoset monkey showed the longest period of 7 hours so far. So this was surprising to me. Initially, I probably naively thought larger species must show longer biological time, longer segmentation clock periods. But apparently, I was wrong. And as you see, the rhino and cow are much heavier than human, but they showed shorter oscillation period. And marmoset is a very small primate, but it showed the longest period of segmentation clock so far. So there's no correlation between the segmentation clock periods and then adult animal body weights. So what's the rule here? Is there any pattern here? Actually, if we take a look at the phylogenetic tree, and the pattern is relatively clear. So we primates, the human and marmoset monkey, showed the longest period of 5 to 7 hours. And the gliales, the mouse and rabbit, showed the shortest period. Then the ungulates were the intermediate. The rhino and then cow were the intermediate. So I can probably say phylogenetically closer species tend to show similar oscillation periods of the segmentation clock, even though I cannot explain why we primates are slowest. But at least this is not random, and this is according to a phylogenetic tree. But anyway, so I was saying that if we compare human and mouse cells, and those reactions were slower in human cells. So what about other 4 species? So we measured these kinetic parameters in 6 species, and then they were perfectly well correlated with the segmentation clock period. For example, this is the degradation rate of Hess7, and this parameter was correlated very well with the segmentation clock period. And on the right is the delay due to Hess7 intron splicing. This parameter was also perfectly correlated with the segmentation clock period across 6 species. So based on these data, we now think that these animals have species-specific segmentation clock periods because they have species-specific reaction kinetics. Protein degradation, transcription, splicing, translation, they are all faster in gliales, the mouse and rabbit, and then slower in primates. So, okay, to address the first question, this slower kinetics in human and the mouse, this concept is generalizable to 6 mammalian species so far. There's a very nice scaling in the biochemical reaction speed. Okay, then what about other genes? So far, we've been focusing only on one gene, this Hess7, right? But there are 20, 30,000 genes in our body, so what about those? That's why we've been systematically measuring the protein degradation rates of all genes, and in collaboration with Henrik Hammeren and then Misha Savitsky's group, by using isotope labeling and then mass spec measurements. So in this graph, each dot represents each gene, and there are about 4,000 genes in this graph. And then the x-axis is the protein degradation rates, half-lives, measured in human segmentation clock cells. And the y-axis is the half-lives measured in mouse segmentation clock cells. And here's y equal x-axis. And compared with this line, the entire distribution is shifted to the right, meaning that there's a global tendency that human cells show slower degradation rates than mouse cells. So actually, among the 4,000 genes we measured, about 1,700 genes showed statistically significantly slower degradation rates in human cells as compared to mouse cells. By contrast, only 126 genes showed significantly faster degradation in human cells as compared to mouse. I'm not saying that all genes are slower in humans. So there are a lot of genes that didn't show significant difference between mice and humans, and then there are exception genes that showed faster degradation in human cells. So it's not all genes, but as a global trend, as a global tendency, human cells show slower protein degradations than mouse cells. And this global trend of slower degradation in human cells can be observed in any types of proteins. So here we classified all proteins according to their localizations. And proteins that, in any category, those proteins showed slower average protein degradation rate. For example, proteins localizing in the lysosome or mitochondria, EL, or nucleus, they all showed slower degradation in human cells as compared to mouse cells. And to verify this mass spec measurement, we performed some independent experiments. So here we made several localization reporters. So this is a nuclear reporter that localizes in the nucleus, and this reporter was degraded more slowly in human cells as compared to mouse cells. And this is a lysosomal reporter and then ER reporter. They also showed slower degradation in human cells. And interestingly, those reporters, localization reporters, are degraded through different mechanisms. The nuclear reporter is degraded through the ubiquitin proteasome system, while the lysosomal reporter and the ER reporter are degraded through the lysosome autophagy system. So even though the mechanisms, the degradation mechanisms are different, they both showed the slower degradation in human cells. So I'm saying that this is a universal mechanism that human cells show slower degradations overall. And one more interesting finding from this measurement is that we can phenocopy slow human degradation profile by treating mouse cells with a metabolic inhibitor. So here in this graph, y-axis is the mouse cells treated with a metabolic inhibitor. So 2DG is a glycolysis inhibitor. And then now this human-mouse comparison becomes symmetric. Their degradation profiles become really similar. And then the average, median half-life, protein half-life in mouse cells was 24 hours. 37 hours in human, and then 36 hours in the mouse cells treated with the metabolic inhibitor. So this suggests that metabolic inhibition in mouse cells can phenocopy the slow degradation profile of human cells. So metabolism may be important for the interspecies difference. So to wrap up the second question, what about other genes? Yes, and this concept of slow kinetics in human cells can be generalizable to many other genes. I wouldn't say all genes, but there's a global tendency of slow degradation in human cells. Then the last question, what's the cause of this slower kinetics in human cells as compared to mouse cells? So this is a difficult one. So recently, Olivier Porky's group published a really interesting paper demonstrating that metabolic rates are lower in human cells as compared to mouse cells. So they measured the mitochondrial respiration rate and glycolytic rate in mouse and human segmentation clock cells. And then both metabolic rates were lower in human cells. And then the authors also showed that metabolic inhibitions can slow down the segmentation clock period. And my group also tested two metabolic inhibitors. 2DG is a glycolysis inhibitor, and sodium azide is a mitochondrial electron transport chain inhibitor. So both metabolic inhibitors slow down the segmentation clock of mouse. Then we thought maybe metabolism is a global modulator of the species-specific reaction kinetics. Maybe these animals have species-specific ATP turnover rate, and then that dictates a species-specific reaction kinetics, because ATP is important for any biological processes, right? That sounded really simple and attractive. However, it was not so simple, and the truth was uglier than that. So let me explain. So I said these three kinetic parameters are important for the segmentation clock period: degradation rate, production delay, and intron delay. So therefore, we measured these parameters in the presence of metabolic inhibitors, finding that metabolic inhibitions are really selective on these kinetic parameters. For example, the glycolysis inhibitor slowed down His7 protein degradation a lot, while not affecting the intron delay at all. And then it extended the production delay a little bit. By contrast, the other metabolic inhibitor, sodium azide, mitochondrial ETC blocker, didn't affect protein degradation rate or production delay at all, but it extended the intron delay significantly. So metabolic inhibitions really showed selective effects on different biochemical reactions. Just as a comparison, sorry, it's getting a bit complicated, but we also tested the temperature shift as a comparison. So normally we culture all cells at 37 degrees because they are mammalian cells. But if we lower the temperature to 30 degrees, we can slow down the segmentation clock, and we can slow down other reactions as well. So protein degradation was slower, and then intron delay became longer, production delay became longer. So temperature shift can modulate all these reactions simultaneously. So temperature shift is a global modulator of all kinetic parameters. By contrast, metabolic inhibitions are not global modulator. It's a selective modulator of individual kinetic parameters. So even though this global modulator of ATP hypothesis was really attractive, I don't think ATP is the global modulator of species-specific reaction kinetics now. Instead, probably there are multiple metabolic factors that can affect a different reaction kinetics selectively. And then each animal species needs to combine those multiple metabolic factors in a different way to achieve species-specific reaction kinetics. So those animals combine different metabolic factors in a different proportion to achieve the scaling of the reaction kinetics. So, sorry, it's getting a bit complicated. To be honest, the more I study metabolism, the more I get confused. But this is our current working hypothesis. So combinations of multiple metabolic factors leads to achieve species-specific reaction kinetics, and that leads to species-specific segmentation clock periods. Okay, and then finally, let me mention the universality of our approach, because you may think that the segmentation clock sounds like a special mechanism in development. I mean, not many developmental processes show oscillations, right? So what we are studying may be an exception in development. However, we found that the segmentation clock period is very well correlated with the general embryogenesis tempo. So here, let me divide the entire gestation period into two phases: the embryogenesis phase and then fetal growth phase. So in the case of human, the entire gestation period is 9 months, Then embryogenesis is about 2 months, initial 2 months, and then the rest of the pregnancy, 7 months, is the fetal growth period. And the segmentation clock period is very well correlated with the length of embryogenesis. So this means that we can use the segmentation clock as a proxy for the general embryogenesis tempo. So by studying the species-specific segmentation clock periods, we may be able to understand species-specific general embryogenesis tempo. Namely, why is human embryogenesis 2 to 3 times slower than entire mouse development? So that's what we are doing. And to wrap up these parts, we found that several kinetics of human segmentation clock are slower than that of those of mouse segmentation clock. And this concept is generalizable to 6 different mammalian species. There's a very nice scaling between the reaction kinetics and segmentation clock period. And this concept is also generalizable to many other genes. There's a global difference between human and mouse degradation rates. And then the cause, and even though we are still studying and we are still a bit puzzled, we now think that combinations of multiple metabolic factors and achieve species-specific kinetics. Okay, and in the future, I'd like to continue studying several examples of interspecies differences by using our stem cell zoo. And so far, we've been focusing on the segmentation clock, and several lab members are still studying their remaining questions. But other lab members are now working on species-specific heart rates. Why our heart beats about 60 times per minute, while other species show faster or slower heart rates. And other lab members are working on more 3D structures of brain organoids. So they are studying the shape and mechanics and metabolism of brain organoids. And we have other stem cell-based models in the lab. And excitingly, after coming to Dresden, we already found new collaborators for each project. I mean, Dresden is a really easy place to find new collaborators. And for each project, we found new collaborators, and then some collaborations are really, and the direction of those collaborations are really unexpected. So I'm really looking forward to seeing how these new collaborations will develop in Dresden. And finally, let me just end my talk by introducing one spin-off project of the stem cell zoo, which is a cultured beef project. So because we have very nice bovine stem cells, and because the segmentation clock cells are actually precursors of the skeletal muscle, we can make bovine skeletal muscle in vitro. And we can even co-induce other useful cell types from bovine stem cells, like endothelial cells that can form blood vessels. So therefore, we are currently making cultured beef steak with blood vessel-like structures. So this is our current mini steak that contains bovine muscle fibers and then endothelial cell networks. And currently, it's really tiny, maybe less than 1 millimeter. And we've just calculated the cost. And one aggregate, one mini steak costs several hundred euros. So I don't think it's feasible to commercialize this immediately. But it's an exciting future, no? Even though my lab is primarily interested in the pure basic science, it will be really rewarding if our science leads to some applications in the future. Good. And then, so I'm of course grateful for the lab members. The work I introduced today was mostly done by Mitsuhiro Matsuda, Jorge Lazaro, Maria Costanzo, and Marina Sanaki Matsumiya. But other lab members are working on other exciting projects. And I'm also super grateful for the support and funding from different institutes and organizations, and of course, Paul. So thank you so much.